Hey, Mr. P here. In this video, we're going to talk about meiosis and compare it to the last couple of videos, which have been mitosis and the mitotic division, the purposes, the structure and function of the cellular structures associated with mitosis. Now we're getting into meiosis. Meiotic division is the production or for the production of gametes, which is sperm cell and egg cells. And so we're going to talk about how these cells are produced, why they're produced the way they are, how the production methods of these cells allow them to be haploid, how those haploid cells differentiate from the diploid somatic cells that are produced via mitotic divisions. And so at the end of this video, hopefully you have an idea of how not only meiosis differs from mitosis, but also the importance of the structure and function of meiosis, and how meiosis is the basis for sexual reproduction. So the first thing that is going to obviously be the difference or a difference between meiosis and mitosis is that meiosis is a reduction division. And by reduction division, I mean we are going to reduce the number of chromosomes in each of our cells. You will notice by looking at meiosis holistically, from the beginning of interphase through the end of telophase, that this cell began with 2 chromosomes. These are unduplicated chromosomes at the moment, and they will be duplicated during S phase, which we'll talk about, but there are 2 chromosomes in this cell. One of them is maternal and one is paternal. Notice one is light blue and one is dark blue. That is to signify the paternal and maternal lineages. And so this chromosome, or this cell, is going to have 2 chromosomes at the beginning. They are going to divide, the number is going to be reduced, and the resulting progeny cells are going to contain 1 chromosome each. That is a reduction division, or required a reduction division, which is one of the purposes of meiosis. Now, because these the number of chromosomes have been reduced, that's going to allow the cells to be haploid, and then when haploid gametes via the sperm cell and egg cell meet, and they're both haploid, then that constitutes a joining of 2 haploids, which become a diploid cell. So, cells in the testes and ovary, called germ cells, undergo meiosis and produce gametes. The gametes, like I've referred to, are the sperm cells and egg cells. Obviously, sperm cells are going to be male, and the egg cells are going to be female. Both of them are haploid, meaning they have 1 complete set of chromosomes, and they have 1 complete set each. Fusion of those 2 haploid gametes in fertilization restores the chromosome number to the diploid number of 46 in human case. Okay, 46 is the human chromosome number. Okay, not every organism or every living organism has 46 chromosomes. Humans have 46 chromosomes. That means our haploid gametes are going to have 23 chromosomes each. So a sperm cell is going to have 23 total chromosomes, and an egg cell is going to have 23 total chromosomes. And when you put the 23 chromosomes of the sperm together with the 23 chromosomes of the egg, you get a viable human somatic cell, okay, or a human zygote in the first cell's case, and that zygote has 46 chromosomes, which makes a viable human. Okay? All of these sperm cells would have 23 chromosomes, or should have 23. Now, there are situations that result in genetic disorders where sperm cells or egg cells do not have 23. In fact, they might have 22 or 24, which would result in a non-normal number of chromosomes that result in our zygote, which can constitute genetic disorders, which we'll talk about a little later. Okay? Meiosis 1, which is this portion of meiosis, reduces the chromosome number from, in this case, 2 to 1. Meiosis 2, on the other hand, which is from here to here, is going to not reduce the number of chromosomes. In fact, we start meiosis 2, in this case, in this particular image, with 1 chromosome, and we end with 1. Now, the difference is we have a full X here and a half X. So we are reducing the content of DNA, or the amount of DNA, but we're not actually reducing the number of chromosomes in the cell. Okay? In this particular cell, we have a diploid cell, 2n equals 2. We get 2 haploid cells here, which is n equals 1. And we end with n equals 1, where meiosis 1 is going to reduce the chromosome number, meiosis 2 does not. Okay? So, let's focus on what meiosis 1 really means, or what meiosis 1 is going to do. Oops. Meiosis 1 in any meiotic division is going to take the number of unduplicated chromosomes in interphase, and it's going to duplicate them in S phase. Before entering prophase 1, the chromosome content needs to duplicate. Okay, so we have two chromosomes. We have 2n equals 2 in this particular case, but they are unduplicated chromosomes. So what has to happen is DNA replication has to duplicate those chromatids, okay, or those unduplicated chromosomes, and actually make the X-shaped chromosomes. Now you'll see the chromosomes that enter prophase 1 are going to be duplicated, and now they are full X's. Again, we still have a paternal chromosome and a maternal chromosome, meaning a chromosome from mom and a chromosome from dad. Now, in this picture, we have one from mom and one from dad. If this was an actual diagram of human meiosis, we would actually have 46 total chromosomes. We would have 23 light blue chromosomes and 23 dark blue chromosomes. But for the purposes of simplifying this image, we just are going to look at one each. Prophase 1, the nuclear envelope, just like we went through in meiosis, in the mitosis videos, the nuclear envelope dissolves, the spindle fibers are formed, these centrioles are going to migrate, but the big difference between mitosis and meiosis is this step, okay? The recombination step. We call this crossing over, okay? And crossing over can only happen during meiosis, and it only happens during prophase 1. Now, what is crossing over? We're going to talk more about it later, but crossing over is when homologous chromosomes actually line up in a tetrad during a process called synapsis, and they swap portions of their arms so that now our light blue chromosome contains dark blue sections, and our dark blue chromosome contains light blue sections. That is called recombination, and it occurs during meiosis 1, specifically prophase 1. Once recombination occurs, the metaphase phase is going to be categorized or characterized by the chromosomes lining up in their tetrads along the equator. And then anaphase, those homologous chromosomes are actually going to separate, whereas one goes to the right, one goes to the left, and eventually you have a cytokinesis event, and one cell becomes two haploid cells at this point. Each of these cells are going to be haploid, and now they are ready to go through meiosis 2. Okay, so what happens during meiosis 2? Both of these haploid cells, which we just produced during meiosis 1, which have been recombined, okay, crossing over occurred during prophase 1 of meiosis 1, are going to go through their own PMAT, prophase, metaphase, anaphase, telophase. They're going to line up in the middle, and then they're going to separate the sister chromatids at the centromere. And so now from one full duplicated chromosome, we actually now have two unduplicated chromosomes, one in each of the cells. Cytokinesis will take place, and now we have 4 genetically unique cells which result from our meiosis event. Okay, all of these are going to be daughter cells, but all of these cells are going to be what we call gametes, which in male's case would be sperm, in female's case would be eggs. You will also notice that because of our recombining or our recombination step up in prophase 1 of meiosis 1 and the crossing over that took place, all of these individual gametes that were produced are genetically different and would result in a genetically unique offspring if each one of these was to successfully fertilize an egg, if these were the sperm cells. Okay? Very important. So, genetic recombination occurs during meiosis 1. This is a slightly different image, but it is going to highlight the same process that occurred in that first image. This would be possibility 1. This would be possibility 2. All of the chromosomes in possibility 1 and possibility 2 are identical. Okay, they have the same gene locations, they have the same maternal group and the same paternal group. We have 2 chromosomes that are red and 2 chromosomes that are blue. Possibility 1 just says based on just random assortment or random arrangement of these chromosomes. Possibility 1 means that both blue, or is depicting an example in which both blue line up on the left and both red line up on the right. In possibility 2, we have the blue chromosome number 1 on the left and the red chromosome number 1 on the right. And our chromosome number 2 pair switched, right? So now we have one blue on the left and one blue on the right, one red on the right and one red on the left. When these go through their meiotic 1 and meiosis 2, or their meiosis division, they're essentially going to split and result in these two combinations, right? Because we didn't cross over and because we didn't randomly assort these the other way like we did in possibility 2, they're going to result in combination 1 or combination 2. Whereas if we line up the chromosomes this way, now when we split in meiosis 1 and then split in meiosis 2, we get this combination and this combination. Now we can further diversify our combinations if crossing over took place. If crossing over swapped the genes on these two homologous chromosomes, now all of a sudden these two combinations would be completely different, and so we would actually have 4 possible combinations as the result of one crossing over event. Okay? Random assortment of chromosomes in meiosis means how they line up physically. Do they line up with all blue on the left, or do they line up with all red on the right? Do they line up with some blue on the left, some blue on the right? Okay, those are the random assortment, and that's independent assortment associated with meiosis. Human cells have 23 pairs of chromosomes. We've talked about that. So that means 2 to the 23rd power. There are 8,300,000 different combinations of maternal and paternal chromosome combinations that are possible, meaning instead of just possibility 1 and possibility 2, there is 8.4 almost combinations or possibilities for all of our 46 chromosomes to line up, which just goes to show you that that's why there's so much variation that exists among people on the planet. Okay, so what does this crossing over really look like? Crossing over is when homologous chromosomes come together, form a tetrad during a process called synapsis. That is called a tetrad. So let's just say that this is the paternal chromosome, this is the maternal chromosome. They come together during a process called synapsis. It forms a tetrad. And then they overlap their inside arms and swap genes, and that ensures genetic variation or even increases genetic variation because once they exchange genetic material during a process called crossing over, non-sister chromatids overlap and get swapped between homologous chromosomes, changing the genetic outcome of the gametes and increasing genetic diversity of the species. Now when they divide, this particular chromosome is going to be A, B, C linked. This one's going to be A, B, little c linked. This one's going to be little a, little b, big C linked. And this outside one is going to be little a, little b, little c. All 4 of those combinations are different. And so we have 4 different combinations from one set of homologous chromosomes because of one crossover event. So when the variability generated by crossing over is added to the produced random combination of maternal and paternal chromosomes, the number of different chromosome combinations that a couple can produce in their offspring has been estimated to be 8 times 10 to the 23rd power, which is well over the 8 million combinations generated just from independent assortment alone. Okay? That is a little bit about meiosis, the steps involved in meiosis, the reduction division associated with meiosis, crossing over, and the recombination that affects the genetic diversity of species. If you have any questions, bring those to class. See ya.